to this this webinar of LMFL. My name is Marcello Meldi, and for today I am your exceptional host because our speaker of today is the usual host of the webinar series, who is Francesco Romanò. So let me introduce our speaker of today with the slide that we have here. Francesco is associate professor at ArcelorMittal Lille, and he earned a bachelor and master degree in aerospace engineering at the University of Pisa in Italy in 2010 and 2012. In 2016, he completed his PhD in mechanical engineering at TU Wien in Austria, with a research focus on fluid dynamics, numerical methods, and dynamical systems. During his PhD, he also held the position of university assistant at TU Wien, where he taught a master's course on numerical methods in fluid dynamics for 4 years. After obtaining his PhD, he kept being affiliated to the Institute of Mechanical Engineering and Heat Transfer, always at the TU Wien, as a university assistant and postdoc researcher for 1 year and a half. And during this period, he was mentoring intern, bachelor, master, and PhD students and continuing his teaching activities. In 2017, he moved to the United States, where he was granted a research fellowship at the Department of Biomedical Engineering of the University of Michigan, where he worked until August 2019. And since September 2019, he joined the Department of Fluid Mechanics and Energetics at ArcelorMittal in Lille, where he gave classes of aerodynamics, turbomachinery, heat transfer, and numerical methods. His research topics include mixing cavities, chaos theory, multi-phase flow in microfluidic systems, such as particle-laden and multifluids flow, biological flows, non-Newtonian flows, thermo and soluto capillary flows, stability analysis, mathematical modeling of complex multi-scale systems, and more recently, flow control and turbomachinery. We want to thank you, our speaker, for the talk that he's going to give today. So Francesco, whenever you feel ready, please share your screen and you can begin your presentation. Thank you very much.
Thank you, Marcello, for the presentation. I just shared my screen and I hope you can see it. And now I will just go in full screen mode. So,
yes, it's good.
That's good. Okay, perfect. So welcome to everyone who is attending this webinar. So the talk of today, the talk I'm presenting today is the fluid mechanics of airway closure in the bronchioles. And I want to thank the main collaborators of this study, which I started when I was in US, and I'm in the group of Jim Brockberg, and we collaborate on this topic with Metin Muradoglu, Hideki Fujioka, and Osan Erken. So the topic of today is actually related to, as you can imagine, to the bronchioles, so to very small flows in the lungs, and the flow physics and the actual broader physics which we have in this kind of system is very complicated. And here you have a small picture which is kind of a cartoon which tends to represent the several difficulties we have in modeling such sort of flows, because we have a complex solid mechanics, we have moving boundaries, we have a 3-layer system, so there is serous layer and mucus layer that are liquids and air, and on top of that we have surfactants which change surface tension and create marathon flows. So just to give you a hint of what we are gonna talk about today, and but I will start with a little bit of introduction, with some introduction for who is not really familiar with the system. Just try to derive very simple concepts or very simple order of magnitude estimates of non-dimensional models, which can somehow support the modeling assumptions we are going to deal with all during this presentation. So here you see basically the branching of airways. So there is the windpipe, the trachea, that's typically of the order for the diameter of 1.5 to 2 centimeters. Then it splits in the main bronchi that have a diameter of 0.8 to 1.6 centimeters. And then you get lobar bronchi, tertiary bronchi, and at the end, where the actual transfer of oxygen is done between the lungs and the blood, you have the alveoli. But what we are very interested in is what are interesting in this presentation are the bronchioles. And for the size of the bronchioles, morphological papers, they report a correlation which is valid up to generation 17. So after you split 17 times from the trachea, and such a correlation is a power law with a base 2. And n in this case is the number of bifurcation, and d0 refers to the diameter of the trachea. So if you want to use such a sort of power law for estimating how large the diameter of a bronchioles will be given a certain generation n, what you can say is you start with the trachea between 1.5 and 2 centimeters, and then you apply such a relation until you end up with, for example, generation 10 that is a little bit larger in diameter than 1.3 millimeters. So you can use such sort of estimate to carry out an order of magnitude analysis on the bond number. And in this sense, you can consider, of course, the gravity, the density of the liquid, and the density of the gas. The density of liquid is basically the mucus, so it's very close to the density of water. And density of gas is density of air, of course. And here you have sigma 0, that's a mean surface tension or reference surface tension, which changes depending on the case you are considering. For healthy cases, we are considering surface tensions which are about between 3 and 2 times less than between water and air, so like 20 dynes per centimeter. And on the other hand, for patients which are surfactant deficient, the surface tension increase up to 50 more or less. So you can have a surface tension range which can vary, which can double or even be multiplied by a factor of 2.5. So that's why we consider several cases. We consider healthy patients which have low surface tension, and we consider small trachea and large trachea to have an estimate of the radius. And sigma 0 can vary. We consider it varying it between 20 and 50 dynes per centimeter. So at the end, if you use the correlation we said before, and you use such sort of interpretation of the data, you end up with a bond number which is estimated here in the plot, and usually assumed if the bond number is smaller than 0.3, you can neglect gravitational effects. And this is why we want to go for bronchioles, because this allows us to have a simplified model for our system in which we can neglect gravity more or less from generation 7 onward. So the problem, of course, is quite complicated by the fact that the bronchioles or the airways in general, they change size because we are breathing, and the walls of the bronchioles basically shrink, and they increase their cross section depending on which phase of breathing we are considering. And in a normal inspiration-expiration cycle, the breathing cycle you see here in green and lila, in a healthy patient, usually you don't observe airway closure. So you don't observe the main phenomenon we are going to deal with today, which I will explain it in a slide. But when you have deep breathing, that's what we are going to discuss in the next slide. you can observe what is called airway closure, so the occlusion of the airways, the occlusion usually in this case of the bronchioles. So what you actually have is that when you are breathing, and here we are considering deep breathing, for example, you can always set an average radius of, for example, of a bronchioles of generation 8, just to give you an example. And then, depending on where you are in the breathing cycle, your lungs start growing in size because you actually are inspiring, and you pass from the blue that's in from the blue marker that's in this condition where the bronchioles are small but they are inflating, to the yellow where the bronchioles inflated beyond the average size of the radius, up to the point that's in green, where the volume of your lungs is the deepest. So when you breathe in very deeply, you go to the green spot, so to the green picture here, and the size of your bronchioles radius is the largest in this easy sketch, in this simplifying sketch. And then you start breathing out, and you pass to the purple that you see here, where the size of the bronchioles is still larger than the average one, but it reduced with respect to before. And you arrive to the red one, where the size of your bronchioles is the minimum. And so the radius of your bronchioles is much smaller, or even half of the average radius. Now, and here comes the why we need to consider such an effect, because it can induce an instability in your system, because what you have is a bronchioles which has a certain length L and a certain radius A. Once again, the radius A can change depending on which phase of your breathing cycle are you in. And there is a certain H, that's the thickness of the liquid coating the bronchioles, which basically is not affected by the breathing cycle, or almost not affected by the breathing cycle. This is meaning that when you breathe in and out, the ratio between the thickness of the bronchioles and the radius of the bronchioles changes inversely proportional to the breathing cycle, or inversely proportional to the volume of your lungs. Now, there is a very classical instability, that's a Plateau-Rayleigh instability, which is driven by surface tension. This instability is actually triggered by surface tension on the liquid, on the mucus air interface, and it's due to the fact that your bronchioles has a cross section which is circular. So there, on this interface, you have a planar curvature which tends to stabilize the phenomenon, so the Rayleigh Plateau instability. But you also have a cross-sectional curvature which tends to destabilize the system, and such a curvature is actually the radius measured from the center line to the interface, and you find it here in blue. A minus H is actually the cross section, the radius of the cross section of the lined portion. So if the ratio H over A changes, so if A decreases, reduces with respect to previous phase of the breathing cycle, you may be more prone to run into the Plateau-Rayleigh instability. So when the ratio between H and A is typical values of the order of 0.1 or 0.12 or 13, you may run into the Plateau-Rayleigh instability, which will tend to close your airway. And this is actually the fluid dynamics that we are interested in, motivated by physiology. Just let me quickly stop my screen share and start sharing a video. So there we go. Okay. You should see my video in a second. Please let me know if you see it.
Yes, it's there.
Yeah, it's good.
Okay, so this is just a description, for the moment, it's just to demonstrate the airway closure. And so here what we have is a liquid film that's a single layer liquid. And at the bottom we have air. And the interface is marked in black, and you find the arrows on the indicating that actually the velocities. And on the bottom plot, on the bottom panel, what you have is the pressure along the wall and the shear stress along the wall. I will enter into details why we need them, but for the moment, let's just show what is the phenomenon. So you wait a little bit, there is surface tension on the interface, you do nothing, you have periodic boundary conditions on the left and on the right, you have a wall at the top, you have axisymmetric condition at the bottom, you just wait the instability to develop by itself, and you see that there is a bulge which is forming and it's draining liquid from the thin film you have near the wall, and that's actually a production of the Plateau-Rayleigh instability. And the more you drain, there you go, the more you you make the bulge growing until you arrive at airway closure. That's actually airway closure already. So you close the airway. There is no anymore gas exchange due to the fact that you are forming a liquid plug, which at a certain point it's relaxed to a certain given shape because we don't have airflow. We can safely simulate it without airflow in this case. And you form this green part here that's a liquid plug, and on the left and on the right you have two air fingers. As you could see, I will show you the last phase once again. The process is growing very fast, actually almost exponentially fast. And the stress, they change a lot right before or right after airway closure. This will be very important for the presentation. So I stop sharing this and I start sharing my presentation once again. There we go. So the airway closure, what we just saw, so the Plateau-Rayleigh instability which developed has been tested experimentally. Its effect has been tested to characterize how important it can be for the epithelium, so for the epithelial cells which are coating the airways. And what you see here is on the left what happens to the cells if they are experiencing a liquid plug which is sitting there. And on the right, if the liquid plug gets ruptured, that's basically the inverse process of forming a liquid plug, and it has comparable stress. And right before liquid plug rupture, the cells they don't get that much affected if you rupture 10 liquid plugs or 50 liquid plugs or even 100 liquid plugs. The green ones are the cells which survived, and the red one are the cells which didn't make it. On the right side, right downstream of the liquid plug rupture, you also see that the stress level on the cells, just because of the rupture, just because of this sudden capillary stress released on the cells, which is basically, as I said, comparable to the formation of a liquid plug, so same order of magnitude, the cells start dying much faster, right? Because they are subject to the largest stress levels and stress gradient levels. And after 10 propagation and plug ruptures, you see an enormous amount of cells which already died. After 100, almost all the cells, they kind of died. So in this sense, that's why what is the significance of such a study in a physiological context? Airway closure as well as airway reopening may induce lethal or sublethal responses of epithelial cells. So we are interested in the stress. That's why we are so interested in the stress along the walls, because they can damage the epithelium. As we said at the beginning, the problem is quite complex because we have a complex multi-layer structure. We have ciliated surface, we have fluid structure interaction, complex multi-phase flow. Mucus is strongly non-Newtonian. We have 2 layers. There are surfactants. It's way too complicated. We need to simplify. There are studies which focus on cilia. There are studies which focus on free structure interaction. In that case, they usually neglect the non-Newtonian part or the 2-layer flow, the surfactants. We decide to approach the problem from the other side, and we don't consider free structure interaction, even though we are aiming to. We consider, so for this presentation, rigid airway walls. We consider no through flow. That's a good approximation for what has been shown asymptotically at the Reynolds number we consider. So that's this is motivated by literature. We consider periodic boundary conditions as we have no through flow. This is typical of all the models you find for airway closure, and that's a simplification which does not affect much the system, because the liquid lining is actually defined in thickness such that it can be modeled by periodic conditions. What we actually are interested in is complex multi-phase flow, non-Newtonian 2-layer system, and surfactant dynamics. We will split this into different parts. Scientific approach in this presentation is working as follows. We start with a Newtonian single layer which has no surfactant, so it's clean. We try to understand what physics we can understand, we can capture with such a simplified system. Then we keep it clean, so we have no surfactant, still Newtonian, and we add the 2-layer. So we add mucus and serum. Then we remove the 2 layers, we keep the single layer, but we add viscoelasticity, so we don't talk anymore about Newtonian systems. And then we remove non-Newtonian dynamics, and we add surfactants. So we try to use a standalone approach to try to identify which physics belong to which of these classes, and hopefully in future studies we manage to build a model that includes everything together. So this is the kind of slide you will see it all along the more we advance in the presentation. That's just like, there is a reference model, that's the Newtonian model, and whenever I do a change on the Newtonian single phase, I will mark it in blue. On the left, we have the cartoon of the actual picture. On the right, we have the cartoon of the model we use. As you can see, this is a single layer, so serum and mucus, they have been homogenized. The epithelium is modeled as a soil wall. There are no surfactants, so surface tension is constant. We use a single field approach solved in Basilisk to, in this case in Basilisk, to come up with a numerical solution. So all what you see in this presentation is simulated numerically. And here at the bottom, you find the non-dimensional groups, which are the Laplace number, the ratio between the gas and liquid density, the ratio between gas and mucus viscosity, the aspect ratio, so the length over the radius of our system, that's of the order of 6 because of physiological reasons, and epsilon is the thickness of the initial thickness of the film normalized with the radius of the system, of the bronchioles. We somehow try to compare our numerical prediction with experimental evidence of the dynamics of coalescence and the stress along the walls, and we kind of match quite well with experiments carried out independently in 2010. So we are confident that the code is capable of capturing the 2 aspects which are important for this study, namely the topological change of the interface and the stress along the wall for what we can. I mean, we compare with the experimental data which are available in literature. But what we also see in this system is that right after closure, so here marked with TC, this is closure time, right after closure, what you see is that there is a sharp peak of the stress that's similar to what you observed before in the video, where for wall stress, this is the peak of the wall stress, so the maximum of the wall stress. And if we want to go a little bit further in the analysis, what we see is that we can correlate the dynamics after closure with the stress, because R min is actually the minimum radius of the interface. So when R min goes to 0, the interface has a radial location, has a minimum radial location which touched the axis, and so airway closure occurred. So it's between this time and that time airway closure actually occurred. But after it occurred, there is still draining from the thin film to the liquid plug, which makes the liquid plug growing very fast. And such a growing, it can be imagined as two air fingers retracting, receding. This is a classical problem of Bretherton bubbles, which either advance or retract. It depends in which case you are, you get a different profile. In this case, we have two air fingers retracting, and this generates different mechanisms for stress. One mechanism is the sharp stress at the wall related to shear stress and shear stress gradients. The other mechanism is related to pressure gradients, which are more correlated to which are actually due to the capillary wave you see at the interface, which we will enter into right in a slide. Just to wrap up the message for this slide, you have a bifrontal plug growth. So there is a liquid plug, which is growing on two fronts, and that's basically the process related to the two air fingers receding, which creates a sharp peak of the stress on the walls. As I said before, to understand the growing of the pressure gradient along the wall, what we need to do is to consider, we can correlate it actually, that's due to the capillary wave on developing on the interface after closure. And as the interface gets closer and closer to the wall, you develop a capillary wave which propagates along the wall, and the propagation makes the pressure gradient growing after coalescence. So the pressure gradient is not related to the sharp velocities, so to the fast closure, but it's rather related to the advancing propagating capillary wave. This is just a picture which kind of tends to summarize what we see. These are all the phases of the airway closure. Here we have the stress levels and the pressure gradient levels, which, as you can see, they are the highest far after closure, because closure occurs between the non-dimensional time 7:36 and 7:37. And here the sharpest peaks are at 7:45, so they are after closure. And this explains why such sort of phenomena in terms of damaging epithelium has not been observed before, as the first simulation for the whole process with physiological parameter has been done in 2019 by our group. And then we can ask ourselves, okay, now that we kind of understood where the stress are coming from and how the basic Newtonian dynamics is behaving, we can ask ourselves, okay, what is the effect of different parameters? So for example, we can change the film thickness epsilon, and what we find is that if the film thickness is larger, of course, the instability should be triggered faster and should be faster because the bulge can grow faster. On the other hand, the stress in pressure gradient, they grow when the film thickness is the smallest. This is also possible to understand because the pressure gradients, they are the only ones correlated to the capillary wave, and the capillary wave stress is inversely proportional to the average thickness, and that's why you see a pressure gradient which grows when the film thickness reduces. So this is what we can learn from a Newtonian system that's single phase. Now we try to build on it to understand the other effects. So instead of considering the homogeneous system Newtonian without surfactants. We still keep it clean, so without surfactants. We still keep it Newtonian, but this time we consider two-layer system. So there is the serous layer and the mucus layer. And of course, there are two surface tensions, one between mucus and air and one between serous and mucus. What changed in the equation is that this term here is a surface tension term, which will be different depending on where we are solving on. And to solve these equations, we don't use any more basilisk, but we use our from tracking finite difference code. We still have the same parameters of the same non-dimensional parameters of before. So Laplace number, ratio of densities, viscosities, and aspect ratio. But we also have a number of other parameters that are like telling us the relative importance of serum against mucus in terms of surface tension, densities, viscosities, total thickness, and relative thickness. So first story we can learn is, first message basically is the two-layer system has a coalescence which is much faster than the single-layer one. This is something we could expect because basically two-layer system has a pre-wetting film that's a serous layer, and the coalescence or the liquid plug closure occurs more on the mucus layer. So it's as if the mucus layer does not need to stick to the solid wall, but it can slide on the serous layer. This is easy to understand. What we could not predict is that it can be even 6 times faster. Now, most of the predictions done for airway closure, they consider it in terms of the closure time for one layer predictions. This prediction is maybe wrong up to 6 times. That's a problem because you underestimate how frequent the airway closure can occur, because if it's closing within a breathing cycle, the airway closure may actually occur. If the airway closure takes longer than a breathing cycle to develop, it doesn't occur. So that's a major input. And also in terms of stability, the thickness, the relative thickness of mucus impact stability differently than what expected from a single fluid model, which is stabilized by the presence of the wall. And here you basically find a critical parameter for a critical parameter in terms of thickness of the mucus for Plateau-Rayleigh instability. Just to make the point, if there is an instability, if there is the Plateau-Rayleigh instability and you actually drive the system to closure, the stress level is by far larger than when you don't have the instability. So then when you consider the green and red curves. So all the other curves where you get the when you get the Plateau-Rayleigh instability, so airway closure, the stress level is by far larger. So this can damage epithelium, and what you find on the green and in red is much more tamed down, I would say. And we arrived to estimate also a stabilizing parameter for the serum layer in terms of thickness of the serum. So whoever wants to enter a little bit more in details about quantification of such estimates, we have a recent publication in JFM. One more point which is important in the two-layer system is the ratio of viscosities for our system. And apparently the ratio of viscosities does not influence pressure and pressure gradients. This one we can expect because they depend on the capillary phenomenon, the capillary wave, which does not scale with the relative viscosity. On the other hand, in our scaling, of course. On the other hand, relative viscosity between mucus and serum basically has a strong impact on the stress and stress gradients, taming it down when the viscosity is larger. Now, this is what we could learn out of a two-layer system, so a three-phase system. Now we go back to the single-layer system, so two phases, one homogenized liquid layer and air. And this time we add some complexity by including surfactants. So surfactants in our model enters through surface tension and Marangoni stress, what you see here in blue at the bottom. Of course, it needs to be modeled, the transport of surfactants. Once again, for this problem, for such a study, we use the same from tracking find difference code used for the two-layer system. Now, as I said, surface tension depends now on sigma. Sigma is the concentration of surfactant over along the interface, and it produces gradients of surface tension which drive the flow through Marangoni system. So we need to understand how to model that, and typical models include a constitutive equation for the sigma, which depends on the concentration of surfactants on the interface and on a strength parameter, basically elastic parameter, elasticity parameter, beta, that tells you how strong the surfactants are in impacting on the surface tension sigma. Of course, the surfactants must be transported usually only inside the liquid. So you have an advection diffusion equation for C, that's the bulk surfactant concentration. And then there is this term S dot C, that's a source term, coupled with S dot gamma, that's another source term, and basically describes how you transfer surfactant from the bulk to the interface. And on the interface, we use the equation derived by Stone, which describes how surfactants spread over the interface. Of course, the coupling between bulk surfactant C and surface surfactant gamma is only provided by these source and sink terms. Of course, we have a bunch of parameters which have been included by this system. Beta is the strength of surfactants. SC and SCS, they are the Schmidt number in the bulk and on the surface. Ka and Kd, they are the adsorption and desorption coefficients, and K is the penetration depth of the surfactants. So if we want to describe an airway closure when we have surfactants, what we can do is to plot pressure and normalized concentration. So normalized concentration means we take the concentration of surfactant and we divide by the initial concentration of surfactant. So C tilde goes from 0 to 1. And for pressure, we also normalize. We actually take always the same color direction. So on the left, you find the distribution of surfactants. On the right panel, you find the pressure field. When you see it yellow, that's air. And when you see it green, that's basically almost the same kind of grading of green. That's liquid. Now, and also when you see the red line here, that's the thickness of that red line is proportional to the concentration of surfactant on the interface. So the red line is basically our gamma, and the color coding here is the surfactant concentration in the bulk. So as you can see at the beginning, we start with more or less a homogeneous distribution of surfactant over the interface. And then as we drain fluid from the thin film to the bulge, we're also transporting surfactant towards the bulge of our interface. And we remove surfactants from the thin film. We do this process again and again during the Plateau-Rayleigh instability, such that at the end we have a strong concentration of surfactant interfacial surfactant affecting the process via Marangoni stress, because here where you have more surfactants, your surface tension is smaller, and here where you have a larger surfactant, your surface tension is higher, and the Marangoni stress will drive the flow along the surface due to the surface gradient. This process keeps driving the system, contributing to drive the system also after airway closure, because you have a strong inhomogeneity of surfactants on the tip of the air fingers compared to the liquid film, the thin film you have on the rear side of the airway closure. Once again, you find such a strong capillary wave propagating in the system, which can be tamed down somehow by the concentration of surfactant, and also by the fact that having a lot of surfactant reduces the average surface tension. So we try to understand something about the dynamics of the system when we carry out a parametric study, and we try to understand what's the effect of surfactant concentration. Of course, it tends to slow down the process because the surface tension, the average surface tension is reduced. And when we consider initial concentration of surfactant that increases, this slows down the Plateau-Rayleigh instability that relies on surface tension. Marangoni stress, they contribute to slow down the system, but they are minor, they have a minor effect with respect to average surface tension. Same thing we can say when we increase beta. Increasing beta means that the surfactant, the same amount of surfactant, has a bigger impact on the surface tension. So increasing beta means the surfactant is stronger. If the surfactant is stronger, this is also meaning that surface tension, the average surface tension gets reduced. Once again, increasing beta slows down airway closure and lowers down a little bit the stress. Not much, but beta slows down the closure and reduces the stress. That's a message. On the other hand, we can play with the other parameters, so the penetration depth, and we see it has a very minor effect. We change 3 orders of magnitude. Not much is changed. So the penetration depth is not such an important parameter for the physics. Also, the Schmidt numbers, they are not very important. We change of 1 order of magnitude, the bulk and the surface tension Schmidt numbers, they are not so impactful. And same thing for adsorption and desorption coefficients, which tells us that our model for surfactants is quite robust because we are mimicking surfactants with such sort of coefficients, adsorption, desorption, penetration, depth, and so on. And the system doesn't seem to be strongly sensitive to them. Good news, the conclusions drawn thanks to such a model can be quite generalized. And we have 2 important parameters, concentration and strength of the surfactants. Now, if we keep constant all the parameters we have here and we change the surfactant concentration, the initial surfactant concentration in the bulk, and we also change the thickness of the airway closure, what we observe is that we have 2 competing effects. Increasing the thickness, of course, tends to accelerate, to speed up airway closure, as we saw before. That's a Newtonian process. On the other hand, increasing surfactants tends to slow it down. So what we can come out with is what we call a physiological stability criterion. What do I mean by that? There are conditions, as you can see here, with the green, indigo, and purple line, in which the minimum radius of the interface will never touch 0, means this system is unconditionally stable. It's not gonna close. The airway is not gonna close. And if we increase bulk surfactant concentration, also the black one, so epsilon 0.2, is not gonna close. It's not gonna make it. So these systems are, when the minimum radius doesn't touch 0, that's a condition in which the system is unconditionally stable for small perturbations, of course, for the initial relatively small perturbations. On the other hand, when that's a concept of classical stability, of course. On the other hand, we can ask ourselves, how can we apply such a stability concept to a physiological system of interest? And the question actually boils down to, does the instability occur within a breathing cycle? So all what you see here is actually scaled by capillary scaling. So if you change the generation of the lungs you are considering, the critical time that's corresponding to a breathing cycle, so how long it takes to breathe in and breathe out, changes with the generation, because that's it should scale with the radius. So of course you have the same breathing time, but the systems are rescaled. That's the non-dimensional time that's rescaled with the radius. So in this scaling, you will see that at generation 8, your breathing cycle, your critical time for the breathing cycle is represented by TCP8. If you increase the generation, in this scaling, the breathing cycle gives a critical time for closure, which is larger than for generation 8, and so on and so on. So for each generation, you can define in such a scaling how long it will take to breathe in and to breathe out. If your instability occurs after a breathing cycle, after the time of a breathing cycle, basically breathing will destroy the stability process, the instability process, so will destroy the bulge which is needed to close the airway, and the airway will not occur. This is what we call physiological stability. When the airway occurs within a breathing cycle, we mark it as unstable. When the airway doesn't occur within a breathing cycle, we mark it as stable. And that's what we have here as kind of a stability diagram, but that's why it's called, we called physiologically stable, physiologically unstable, because it sets a finite time to define stability. If the airway closure occurs within a breathing cycle, that's meaning that the system is physiologically unstable, and all the open markers here, they are physiologically unstable. And as you can see, changing generation, the impact of the thickness and of the initial surfactant concentration is quite important. And we can drive actually a simple conclusion out of this plot, that if we have therapies for replacing surfactants, so increasing actually C0, you can stabilize the system, because as you can see, the black, the full markers, they gain stability for high, they let us gain stability for high C0. So the higher C0, the more full markers you find. And you can also use surfactant therapies, surfactant replacement therapies, to shift the instability, the occurrence of airway closure, from low generations to higher generations. As you can see here, for low generations, you are more stable than for higher generations. So you can transfer it to higher generations. So that's what we could try to simplify and to learn adding surfactants. Now, the last step we will consider is including a non-Newtonian dynamics. So we don't have any more surfactants. We have a single layer, and we include viscoelasticity through the stress tensor you have here. Now, there are several ways to model it. One way is using, probably one of the simplest way, is using the Oldroyd-B model, but we tested also other models. I will show a comparison about that, but the idea is the new physics can be well captured by the Oldroyd-B model. What we have in this stress tensor, there is a part which is a Newtonian part. That's basically this part of the flow. And on the right side, you have phi, that's a phase field or indicator function, times S, that's the extra stress, and that's the polymeric part of the stress. In this system, we have another non-dimensional parameter that's telling us how the relaxation time of the polymer, modeled more or less as a spring, as a dumbbell spring, compares with the characteristic time of the system, that for us it's the capillary time. And on top, we have two viscosities, solvent viscosity and polymeric viscosity, which end up proposing another parameter, that's the ratio liquid solvent viscosity over total viscosity. This parameter is actually telling us how charged our solution is with polymers. There is also another parameter which is not there in the old RED-V model, but it's there in other constitutive models we used, like the FNSER. That's the L2 parameter. This is telling you how extensible a polymer can be. For the old RED-V case, L2 tends to infinite, so you can extend the polymer as much as you wish. For the FNSER, you limit the extensibility of a polymer. Now, just to have a quick picture of the point we want to make with this analysis, we compare Newtonian and non-Newtonian viscoelastic stress. What you see here in dashed, they are the Newtonian cases, and as you can see for the stress at the wall, there is always a peak and then a relaxation phase. What we have when we include viscoelasticity, there is still a peak. The first part of the dynamics, that's why it's interesting to compare the models. The first part of the dynamics is basically Newtonian. We get the same sort of phenomenon because the first part of the first phase of the airway closure is driven by surface tension. So and it's redistributed inside the system thanks to viscosity and pressure in axial direction. So basically, this is a mechanism which has nothing to do with the extra stress. On the other hand, after closure, what you find is that there is a second peak or a second hump of the stress, which is there when we include viscoelasticity and which is not there when we consider the purely Newtonian system. So what we find is actually a qualitative difference between Newtonian and viscoelastic systems, which we try to understand. But before trying to understand it, let's try to see if it's robust, because we used, as I said, this probably the simplest model for viscoelasticity. Let's see if we can reobserve such a phenomenon when we complicate a little bit the model and we consider FNSER. In this case, what we do is we change the extensibility of the polymer. Changing the extensibility means that the polymer can stretch more, or if the L2 is larger. What we see is that when the L2, when the L square is quite low, so the polymer cannot stretch, we almost lose such a, or we actually lose such a second hump, which points towards a first hint or a kind of a easy conclusion. The phenomenon of the second hump is robust with the viscoelastic model, but it must depend on a stretching of the polymer. So there must be a phenomenon which connects the flow dynamics with the stretching of the polymers. And when the polymer can stretch enough, it can feed back on the system and produce this stress. So what we then can check is the distribution of the extra stress. And I invite you to focus on the stress we have at the top left panel, which are the most important one for the exercise at the wall. What you see here is that there is a very sharp peak in red. You see it here. That's a sharp peak in red, which is preserved in the system, and it's correlated with the surface, with the capillary wave. Actually, it's propagated by the capillary wave. And that's where actually the system produced the highest stress due to a curvature of the streamlines. So there is a curvature of the streamlines which tends to stretch the polymer. The stretching of the polymer feeds back on the flow system because of the extra stress tensor, and it produces a peak of the stress at the wall, actually produced by a peak of the extra stress. So this is a very classic viscoelastic mechanism for instabilities, and it can be present only if there is a curvature, a pronounced curvature of the streamlines, which is here produced by the pronounced curvature of the interface. And then we try to characterize the system based on the Laplace number, the Weissenberg number, and the polymer concentration. When polymer concentration is the lowest, so solvent dynamic is almost the solvent viscosity. Dynamic viscosity is more or less is 90% of the total viscosity. Means low concentration of polymers. We almost don't observe anything. When we have 50-50, so 50% of the dynamic viscosity is coming from polymers, and the other 50% is coming from the solvent, we start observing a pronounced mechanism of instability for which depends on the Weissenberg number. So it depends how slow the polymer reacts with respect to the characteristic time scale of the system, but it also depends on how strong the surface tension is, because it depends on the Laplace number. Even more, we see such a phenomenon when we charge the system with more polymers, which is a little bit stretching the validity of the model, but up to here we are good. So what we can understand is that the instability must depend on the Laplace, on the Weissenberg, on the Laplace number, the Weissenberg number, and on the polymer concentration. And we try to characterize it by considering that the phenomenon is actually a linear phenomenon, so we can trace the growth of the peak of the extra stress, fitting it with an exponential, either in the phase in which it's growing, so giving rise to the instability, to the elasto-inertial instability, either when it's decaying. So we can plot the sigma, so the growth or the decay rate of the of this fits, and we can characterize when actually when the instability occurs. That's what we do in this diagram with the sigma. This time, sorry for the misunderstanding or the bad notation. Sigma here is the growth rate. It's not the surface tension anymore. And what we see is that the instability depends also on the Laplace number, so on the inertial part, but it's strongly related to the Weissenberg number and the polymer concentration, so the elastic part. So we can characterize it as a phenomenon which is elasto-inertial. I just dropped the conclusions because I think I went a little bit too long with my presentation. I just want to acknowledge the support from NIH and TÜBİTAK, which actually gave us the chance to carry out this research. So thank you for your attention, and if you have any questions, I'm happy to reply.
Thank you very much, Francesco, for this very, very interesting talk. And I think that we can have a look if the participants want to open their mic and ask you a question about this research work, or perhaps they want to write a question in the chat. Please feel free to interact. I see Henry Kuhlmann. Yeah, please go on.
I have a question regarding the flow. You said the flow can be neglected in the airways in your analysis. Is that due to the fact that when closure occurs, you have that minimum of the volume of the breathing cycle?
Mm-hmm. That's there are two typical arguments for that. Actually, three. One of them is we are considering the strongest, I would say, is that we are considering distal airways. So it means bronchioles which are far from the trachea. This is meaning that the size of the bronchioles is very small, and the Reynolds number we have, they are really, really small. And on top, we are considering a multi-phase system in which there is air, and mucus can be about from like 10 to 10,000 times more viscous than water, which makes the viscous stress of air almost negligible compared to the driving force, which is surface tension. So in this sense, that's the strongest argument I find.
Yeah. Okay, I see the scale argument. And related to that, is the length of these sections, does it scale the same way as the diameter? You gave the scaling law of the diameter for the different generations.
There are, of course, we are always averaging because it's like biological systems. That's, but there is quite a sort of golden ratio applied to to bronchioles and bronchi. This ratio, the ratio of length over the radius, is strongly it's almost always 6, or kind of there is a very small the average ratio is 6, and then you get a small deviation from that value.
It's almost 2 pi.
It's almost 2 pi. Yes, that's. But it's. Oh, I have one point on this, which may be interesting.
And that's your periodicity length, no?
Yeah, that's we use 6. Yes, yeah, the Plateau-Rayleigh instability. So you know that the classic paper of Rayleigh considers the instability for a length which is technically infinite, and then you get the wavelength, the critical wavelength, which is what I reported here. Now, this wavelength is unfortunately very close. The ratio between the radius and L, it's unfortunately very close to what we have in the lungs. So that's why we see the instability, honestly, because that's very easy to trigger
okay. But maybe that has also certain reason that nature arranged that in this way.
Yes. That's it's only fluid mechanically unfortunate. Yeah, that's there are optimization reasons for the morphology of lungs, of course.
And if I'm allowed to ask another short question, I was wondering, maybe it's not relevant to your case, but I was thinking of whether there is any theoretical analysis near the collapse point in terms of asymptotics, which you could eventually compare with. I don't know if that exists.
Yeah, that's this one.
But maybe it's you cannot resolve that numerically.
Yeah, we are quite close. The asymptotics of the liquid bridge right before closure should go like with a power 1/2. Okay. And that's what we see here. What we find right prior, and this is the 740 is derived by the asymptotics of the thin film.
Okay, great.
We're not too far.
Okay, excellent. Thank you very much.
Yeah, thanks.
Okay, are there any other question? I can see Mr. Barmark open his microphone. Please go on.
Yes, Francesco, you said that you use periodic boundary conditions, right?
So I don't hear it very clearly. Maybe can you
hear me?
Yeah, but it's quite low, the volume.
Okay, okay, I don't know, but hopefully you can hear me. You use periodic boundary conditions.
Yeah.
So it means that you assume that the length is like quite large. Can I do it? And but then you limit it like to 6, like the length.
Yeah. Okay. Yeah. So I mean, we use periodic boundary conditions because that's the that's somehow how to say, this is the classical framework to study the airway closure as Plateau-Rayleigh instability in the like in the respiratory community, respiratory flow community. We don't set it to 2 pi, we set it to 6, so it's not Fourier. Just if the point is coming from numerics, we don't solve by Fourier modes. We really it depends on which code we use. We just enforce periodicity at the instability, at the order of discretization we use. So either finite volumes and finite differences, we either use order 2 or according to what we are discretizing, we know order 5, and I mean the 6 is driven by biology rather than by numerical convenience.
Okay, yeah, I got. But actually, my main question was not about this, but and then you said that like the film thickness epsilon. From one hand, it depends on the breathing cycle, yeah. So it's time dependent. Yeah, like you show the graph like H over A, yeah, like changing the minimum is, yeah, the deep breaths.
This one.
Yeah, this one. So
yeah.
This is actually sort of your like film thickness, how it's changing with time, right?
This is actually, yes, this is the ratio between H and A. Because H, basically the film thickness, almost it doesn't change much. I mean, within a breathing cycle, you keep the thickness you have when it's not fully collapsed, the cross section, that's what you see. The thing which you change is the radius of the airways, and as you change the radius of the airways, so A, which changed like the breathing cycle, so your volume, the volume of your lungs growing or shrinking. So A changes in phase with the volume of your lungs, and so H over A should change in antiphase.
Yeah, but then epsilon is actually H over A.
Yes,
right?
That's epsilon. That's the epsilon.
So it's like your initial, let's say, because epsilon, then it's changing in time, and then it's you actually what you observe that it's sort of monotonic effect. Like the largest is epsilon.
Yeah.
Yeah.
Yeah. This is
faster
so.
is a process like of.
I see your point. This is that's actually a good point, because technically the model we use does not directly correspond to the actual system we have, because we are fixing we are saying that the wall is rigid, and we are fixing the epsilon, which is somehow not consistent with this argument. It's just like, this argument is just to say, let's see what happens with different thicknesses. But I agree with you, we should have we should consider an epsilon that changes with time, but modeling it risks to include artifacts in the system which are in the model which are difficult to predict the effect of. What people do when they want to consider this is they actually include deformability of the walls.
Yeah,
so this is what it's actually
what my, so in fact, like you have like in reality it's like deformable, so it's okay. But because I think that when you're talking about your time scales, maybe this is sort of your upper bound, yeah, like because you just assume, okay, this is my maximal like breathing cycle. Okay, so
okay. That's but it's a good point. It's a good point. This is like, of course, that's model prediction, but as I said, it should. Sorry. Yeah, that's what fluid structure interaction, what can somehow bridge the gap. I totally agree with you that in this kind of systems, modeling can be used so far to understand the physics and to predict the direction, what is important, what is not important, doing like I would say order of magnitude analysis or understand the physics, predict a certain physics because certain instabilities we observe, we are checking if we find them in experiments. But I agree with you, arriving at a predictive model, which can must include many of such effects, which we are trying to build on it, but there is a long way to go. Yeah.
Okay, got it. Thank you.
Yeah.
So we have received actually a question in the chat by Raja Lakaraju. The question is there a possibility of a traveling wave with a sudden strong shock, either for single or double layer model, and also symmetry breaking at top and bottom layers?
Yeah, question is very good. Of course, that's so break of symmetry. Our argument is let's do an order of magnitude analysis such that we don't need to consider the break of symmetry, but of course gravity is symmetry breaking effect, and when you lay down on a bed and I would I mean, I would hardly think that your airways, they are nicely
yeah, but they are very small, to be honest. But they are very small, so gravity effect probably is not so critical.
Yeah,
and it's like very small diameter.
And yeah, and so gravity actually it's a symmetry breaking effect. When possibility of traveling wave, so there is possibility of traveling wave that are actually, well, there is possibility actually of capillary waves. So if you consider, and this is studied in literature actually, for they do it with rigid pipes, but you can do it with this system as well. You will find it. It's basically if you consider gravity in the axial direction, what you find is when you have very thin films, which is not the case in our system, because thin films in that case they drive strong capillary waves. In our system, we have quite thick films because we want to arrive at airway closure. In that case, you may have capillary waves traveling on the surface of the mucus layer. Strong shock. That's this is I am not aware of any study who investigate shock in for physiological relevant parameters in such a configuration, though there is in a simple configuration, though there is a shock wave which propagates when you have surfactant-laden layers. And in that case, basically you find I think mathematically it's classified as a degenerate shock wave because maybe it's related to dispersion or whatever. But there is a shock wave related to surfactant-laden flows. Also symmetry breaking at top and bottom layers. Symmetry breaking can also, I didn't mention it, but what we found is also like at the bottom layer, you can have a second breakup of the thin film, which if you consider a non-symmetric system, you can basically have also symmetry breaking inside the two layers. I hope that was okay.
Yeah, the researcher thanked you on the chat, Francesco. I don't know if there are any other questions. I kind of have a short one. It's a wee bit about something that you have mentioned rapidly in the presentation in your second answer. It's about fluid structure interaction, and in particular about the cilia, because the cilia are a very interesting mechanism, and they are everywhere, you know. Can you just give a wee bit of detail of perspective of where we can go with the modeling, including this kind of elements?
Yeah, so that's so the cilia are. I just do a very quick point in terms of physiological relevance. In the lungs, cilia are used to move or redistribute mucus. So they have an active effect on they have an active impact on respiratory flows, and they have kind of protecting from pollutants, because basically you tend to move upstream up to the windpipe the excess of mucus or the polluted mucus thanks to the cilia. So basically what they, at least this is my understanding of it, and that's very basic. What they actually have been modeled with is a leading order approach that's basically a bulk force, which moves periodic with a certain period or a certain frequency or a certain coordination among cilia, drives the mucus or mucus and serous layer. The problem I see in this kind of system is that if you want to properly model the cilia, you need to include the fact that they can have collective dynamics, so they can kind of coordinate to. Because if you want to transport mucus upstream, you need a wave. Okay, so the cilia kind of tend to coordinate pushing the mucus in an efficient way without oscillating against each other, or without there is like a collective dynamics of the cilia. Second challenge which I see, which is highly non-trivial, at least for the kind of modeling we are using. Our model is axisymmetric, and the cilia are far from being axisymmetric. So that's if you want to go for modeling them, you need to generalize to fully 3D. And this is also related to fluid structure interaction. Fluid structure interaction is not just squeezing the airway in radial direction, it's also considering the fact that the airway walls can have a structural instability, which is actually observed and even modeled numerically. So you run into a strongly three-dimensional system. That's a challenge to include such a complexity, and on top the complexity of the flow, which is enormous. So, yeah.
Okay, thank you very much, Francesco. I see that there's another question from Joran. Please, Joran, go on.
It was just something that came to my mind when you talk about the cilia. I also suppose that there must be wetting effects on each cilia of the two layers of fluid.
Yeah.
That may kind of change the. So if you start having bridging the two fluid layers and closing the airway, I would suppose that we would deplete fluid elsewhere, and then you would try to de-wet on the cilia, and maybe the cilia would kind of resist the de-wetting, or.
Yeah, that's this is actually a good point. Actually, the problem with the cilia is that they are biologically optimized for healthy conditions, but when you have pathological conditions, the thickness of mucus can pass from like 2 to 4% of the airway radius, up to 40%. So wettability is definitely a problem, because up to a certain point you don't cover the cilia, and beyond, if you're, I mean, the cilia cannot operate anymore as efficiently, because they are fully immersed into mucus, and they need to move like a very thick layer of mucus, which is unpractical for and wettability it is a point because cilia, they get in contact with two different liquids which have different wettability, even if they are both water-based. So there is a serous layer, which is basically waterish liquid with a very little polymers, so it's weakly viscoelastic, and the mucus layer, which is strongly non-Newtonian, which it's overloaded of polymers. Wettability becomes not a trivial effect. I agree.
Okay, thanks.
Yeah.
Okay, do we have other questions for Francesco? No? So, well, possibly, what do you think, Francesco? We can stop here for today. Is it okay?
Yeah.
So thank you very much to our speaker for presenting this very, very interesting research work. And thanks also to everybody that participated and enriched the discussion with very interesting questions.